You folks are in for a wonderful talk, so you should be pleased to be here. I'm very honored to introduce Dr. Kristi Anseth for this keynote address. I strongly recommend that you take a look at your program book, which has a more complete bio than I'm going to give. So please take a look. She's really one of our outstanding researchers. If you were at Leroy Hood's talk yesterday, he talked about paradigm shifts. And Dr. Anseth has been a world leader in another paradigm shift in the area of implantable biomaterials. This area has been dominated by borrowing of materials from other areas. I, for instance, have a titanium artificial hip, which makes me feel like part of an aircraft. And Dr. Anseth realized that it was time to deliberately design materials de novo for use in the body. And in particular, her area has been in the area of in situ photopolymerization, macromolecular constructs that include many different kinds of biomimetic properties. And it's led to a generation of much more effective implantable materials with applications in cartilage replacement, heart valve, tissue engineering, microfluidic bioassays, tissue adhesives, and much, much more. And I think we're going to hear about some of those things. So once you understand that she has been the leader in this paradigm shift, you immediately understand why she has multiple named professorships after her name, and she has one fewer than, I believe it's 20 people who has been elected to all 3 national academies, engineering, science, and the Institute of Medicine. Her brilliant work and leadership have led her to becoming the first engineer to be named a Howard Hughes Medical Institute investigator, and she has also won the Alan Waterman Award from the National Science Foundations. Her successes and awards, and I've constantly heard from her students that have gone off to other walks of life who give her accolades. They're all extremely well-deserved. So I'm extremely pleased to introduce Dr. Kristi Anseth for our keynote talk. Thank you.
Well, thank you for the kind introduction, and it's certainly quite an honor to have this time to tell you a little bit about some of the way that we think about designing new types of biomaterials and working in this field of tissue engineering or regenerative medicine. And so what I'd like to do is walk you through a little bit of a story today of where we began in this field and some of how that thinking has evolved and directions that we think about for the future. But at first, I'd like to begin with a little bit of motivation. So using cells within our body to get it to heal itself when processes go awry is an area that's rapidly expanding. It may not necessarily be such a new concept. Many of you may have had blood transfusions, or you may have known somebody who's had a bone marrow transplant to treat certain types of cancers. But growing in this list are things like the Edmonton protocol, where islets are transplanted from a donor to a patient who has severe diabetes that can no longer be regulated by insulin injections. We'll talk a little bit about autologous chondrocyte implantation. That's taking your own cartilage cells and injecting them into damaged joints to try to coax regeneration of cartilage tissue. And in fact, if you look at even just particular types of cells like stem cells or progenitor cells and do a search on the FDA website, you'll see thousands of clinical trials using cells, injecting them into patients' bodies with the hopes to alleviate symptoms or devastating effects of disease. Now, despite this growing list and going from injecting cells into patients' hearts after a heart attack to neurons into patients' brains with Parkinson's disease, while this is all growing, many of the treatments are suboptimal. And what happens is when you just deliver the cells to the body, in many cases you get what's called low engraftment. So very few of the cells survive, and of those that survive, they may have a limited functional capacity. Now, it doesn't mean that it can't still have tremendous benefit, but it makes you begin to think about how might we improve upon this and get cells to survive, to integrate, and to function. And from that perspective, we think about our bodies as containing not just the cells as building blocks, but they all reside in a material environment, a niche. And that material gives them many different signals that are important. Now, when we think about designing a material as a cell carrier, there are lots of different materials one could think of. And today, I'm going to tell you about one type of material that we've been working with, and that's a hydrogel. And this is just a macroscopic picture of a hydrogel. If you could see the molecular level details of this gel, what you would see would be long macromolecular chains that normally would just dissolve in water. But because of interactions between these chains, it renders them insoluble. So instead, they imbibe large amounts of water, and that gives them some very unique properties, especially unique properties for embedding cells inside of here. And so here I'm showing a picture where we fluorescently labeled the cells so you can see them here. And they can be surviving and living inside of this environment. And in part, it's because of the high water content, which allows for facile transport of important molecules for cells to survive. And it also imparts some unique properties of many of our soft tissues. Our body is comprised of large amounts of water. So where does the engineering come into some of this? So we think about this, and once you put cells in these types of environments, the challenge becomes figuring out how do cells get signals from that environment? Which signals should I include? Over what time scales, over what size scales, from the nanometer scale of a cell binding to something all the way to micron scales where cells come together to something that might be a tissue subunit that could be several millimeters or centimeters. So this is one way that we think about these hydrogel scaffolds, and I won't talk too much about chemistry, but there are many, many different types of hydrogels that one can select. And so one that we've worked a lot with is a material called polyethylene glycol, or PEG. Now, PEG is a synthetic material that we can engineer to have lots of different properties, and it's widely used in human medicine. And one of the reasons is that PEG is very hydrophilic. It has all these waters of hydration around it. And this renders them quite useful for making hydrogel systems, and ones that are relatively inert from the cell's perspective and from the body's perspective. And so when we encapsulate cells inside of these environments, you'll see here, these are chondrocytes, cartilage cells, taking on a spherical morphology. The particular chemistry, here, this is what polyethylene glycol looks like. It's repeating structure. And we modify the ends with different reactive groups so that when I dissolve this molecule in the presence of cells, I can catalyze a reaction. And oftentimes we use photochemical initiation of these reactions. And this linear liquid solution will convert to a solid gel, all in the presence of cells. And you can think about this mesh structure that results as what really controls a lot of the properties of the gel. Now, another way that we think about these hydrogel environments and using them for cell delivery is, on the one hand, we design these systems to be completely permissive. We design hydrogels where the cells are primarily getting a physical or mechanical cue or three-dimensional registry, but the cells don't recognize this chemistry. The cells instead are doing all of the work of making and decorating this permitting environment and regrowing tissues. So I'll start part of today's talk with talking about an example of a permissive gel formulation. But in many instances, one also needs to think about promoting cells. Perhaps we need cells to proliferate or differentiate into a certain cell type, or secrete and make a tissue at a faster rate. In promoting gels, we began to include different biochemical cues that can signal to the cells and signal in a way that we can hopefully manipulate the proper functions that we would like. So let's begin with the story of some of these permissive gels. So when we began some of our work in this area about a decade ago, we became interested in the problem of how might we restore cartilage or resurface joints where joints are diseased or have damaged cartilage. So cartilage is an interesting tissue. It's found on all the ends of our articulating joints. It provides cushioning and allows lubrication and movement without pain. But unfortunately, cartilage has a limited capacity to heal itself. And when it becomes damaged, that damage can lead to further erosion of the tissue, and you can end up with an osteoarthritic joint where you eventually have bone on bone, and it becomes quite painful to move that joint. So right now, the clinical strategy would be to have a knee replacement. So the bone and the underlying tissue, or the cartilage tissue interface, would be removed and replaced by a synthetic replacement part. Now that can be okay if you're older in age when this happens, but the problem is that if you're young and have an arthritic joint, that it won't last your lifetime, and it also limits some of your activities. So when we began working in this field, autologous chondrocyte implantation was starting to come out and be marketed. The idea here is that we all have extra cartilage tissue in our body, and those cells are responsible for making the cartilage matrix and turning it over our entire lifetime. Now, autologous chondrocyte implantation takes a biopsy of your own tissue, collects the cells, the chondrocytes from that, and then tries to grow them up to create a clinically useful number of cells that could then be injected back into one of these damaged knee joints with the hopes that those cells could regrow a tissue. They already know how to do this. Maybe I just need to put cells into this. Now the problem with this, though, is that this procedure, when it was first coming out, worked somewhat for small defects in cartilage, but not for something that would be a really large defect or something where you'd have lots of wear in the joint. And so we became interested, well, could this work better if we had a carrier for these cells and you didn't just inject the cells themselves? So this was our idea. If you take a piece of cartilage tissue and you section it really thin, and then you stain it, this is what it looks like, where these are the cells, these chondrocytes, and those chondrocytes are secreting and making the cartilage tissue, which is comprised mostly of collagen fibers. It's a special type of collagen, type 2. And these are kind of like the mechanical reinforcements or struts. And then they also make these molecules, these proteoglycans, where these are highly branched macromolecules that have negative charges on them. They're sulfated disaccharides. And this causes the tissue to imbibe large amounts of water. And it also gives them some really unique mechanical properties when it gets strained to high extents. So we'll be looking at some pictures of this, but the tissue is also a large amount of water. So our idea is, let's use a in situ forming permissive gel to deliver these constructs. So it could just be delivered onto the joint surface and solidified, and we use light to solidify it in vivo. Now, the question becomes then, what chemistries should I use? So very briefly, so this is that same PEG molecule, and then we just graft off of this short little repeat units, and this one happens to be what degradable sutures are made of. It's a lactide repeat unit. So that when we use this type of precursor and react it to form a gel, these units here will hydrolyze and break down in the body over time, so that the gel is not permanent. Instead, it dissolves away as the cells begin to make cartilage matrix. And so this was one of our early gel formulations, and when you encapsulate chondrocytes in them, they take on this spherical morphology, which is very much what they look like in native tissue. And you can control the density of cells in this, and the engineering question now becomes, how do I make this matrix with the right types of properties so that the cells will function properly and maintain this phenotype where that they'll secrete and make collagen and proteoglycans? So from that perspective, when you work with hydrogels, if you remember, you have this three-dimensional mesh in space. So a lot of the properties of these hydrogels are highly coupled, and they all depend upon the strength of these interactions and the number of them. And so for this application, you'd like to have a tissue that has a reasonable elasticity so it can withstand stresses in the joint. But you also have to have a reasonable amount of water so that the cells can survive. And so what happens as you go to more cross-linked gels, the water content goes down, but the elasticity goes up, And kind of in this region is about what native cartilage matrix would be in that zone. So you have kind of this trade-off, but it's a little more complicated than that because the cells are sitting inside of this gel and they're secreting and making these matrix components. So you also have to care about how those matrix components can diffuse through the gel. And that depends, again, on this mesh size and the size of those molecules the cells are making. So we went through in the early stages and we looked at all these different cross-linking densities and we watched the cells make these matrix components. And what you can see here, so this red staining, that's for that proteoglycan molecule that the cells make. And so the cells here are these dark red dots here. They're making lots of proteoglycan, but it's very much diffusion limited, and most of the proteoglycans is only in the paracellular region around the cells. But if you open up your hydrogel mesh size, you can get very good distribution, and this is able to be elaborated throughout the entire cell-laden construct gel. And as engineers, we like to model some of these so we know rates at which cells make these molecules and how it diffuses. And roughly, this is about what this system's like. And then if you change the diffusion coefficient by changing the mesh size, now you get a system that looks more like this. But cartilage has two very important tissue matrix components, and the second is collagen. And collagen gets assembled through a complex series of events where cells make procollagen molecules that need to assemble, and they form these higher cross-linked fibers that are nanometers, hundreds of nanometers in diameter. And if you look at this gel, what happens to the collagen here, again, these are all the little cells here, and the blue is for the collagen. The collagen is so large that it can't assemble even inside of this type of loosely cross-linked gel. And so instead, for collagen to form in tissues, you really have to control breakdown and degradation of your material. And so we've done lots of different modeling of this. So the chemistry just depends on what you pick for that degradable unit. But the degradation is all the statistics of how connected it is and how many connections do I need to cleave before the gel begins to lose mass and open up in structure. And so I'm not going to go into the details of this, but one can predict by understanding the kinetics of how fast these groups hydrolyze, combined with connectivity to predict and understand how the gels lose mass. And so they began to lose these cross-links and open up quite dramatically. And then it reaches kind of a critical point, and at this critical point, that's when you no longer have this gel anymore. You've broken the last few critical links and it just becomes dissolved. And so that's what happens here, kind of the reverse of gelation or the reverse gel point. So you can begin to tune this by controlling connectivity. And here's the system that we were in before where the gel's degrading too slowly. Here you speed it up by having a more loose connections in your network, fewer network connectivities, and the gel dissolves. It reaches this point before the cells have been able to make enough collagen. But if you tune this, you can get this about right to match the rate of production of this by your cells and the time scale for it to all assemble. And now if we fast forward through lots of work, so this was a lot of bench work, trying to design just a simple permissive system that would allow these cells to make tissue. And with a collaboration with Jennifer Alicieff at Hopkins, we've done some work where we've tried to then repair defects in cartilage joints. So this is a goat's knee joint. And if we deliver our gels with chondrocytes, we see large amounts of tissue regenerated after 4 weeks. And you can, again, even look at a section of this and look at what the collagen or what the cartilage looks like. And here's the bone and what native cartilage looks like in the animal's joint that was undamaged and repaired. And here's what we regenerated. So there's even a striking similarity to that of the native tissue. Now, I tell you this because here we started out with this problem with just an obvious cell source. We just wanted to design a simple carrier, permissive carrier for these cells to make tissue. And it takes a lot of time to learn about how these cells make these tissues, how do you get the right properties, how do you test them in an animal joint, and these have also now been treated in a small clinical trial in Europe in humans. But there's still problems that persist. Things of how do we get these cells to make tissue faster? This can work very well sometimes for younger patients, older patients, their cells don't make matrix as fast. So how do we have a single product for everyone? Interfaces become very important. We're still trying to tackle the challenge for long-term stability of this. Can you get cartilage that doesn't just regenerate in this gel, but what happens at the interfaces? And then that's all in the context, too, of this becomes personalized medicine based on a cell-based therapy, which now becomes much more expensive and difficult to market compared to a synthetic replacement knee joint. So those are some of the business side of the challenges, and there's still some technical challenges. So we began to think about this and realizing that perhaps we want to design materials that could perhaps better integrate with surrounding cells in the tissue, or perhaps even be used with something like a progenitor cell that could be banked and not just use an autologous chondrocyte for the transplantation. But the problem is that while permissive gels work really well for chondrocytes, so here remember they take this rounded morphology, that's how they look in the tissue, but these cells, they don't recognize this chemistry at all. And many, many cells in our body, and many, many stem cells and other cells you'd like to use, they get lots of information from their surrounding environment. They're anchorage dependent. They require lots of cues. For example, here's one of these bone marrow cells circulating in our bone marrow that can go on to form a chondrocyte and make cartilage tissue, bone tissue, and several other tissues. If you put these cells inside of a permissive hydrogel, here's what happens. The cells take on this spherical morphology. They're trying to interact with this matrix. They're trying to migrate towards one another, but they can't. The gel is impeding this, and they don't have any type of chemistry in which they can attach to it. And these cells, over the course of several hours to days, are going to undergo anoecus, a programmed cell death because of lack of matrix interactions. These cells need a promoting environment. So one of the things we began to think about is how could we design promoting gels that could perhaps be used to take cells and deliver them, but then in a way that the cells could perhaps disperse and integrate with the surrounding tissue and get signals that would allow them to survive. Or maybe you would just deliver the material itself, and this material would coax or promote local cells to migrate in. And when they migrate in, they could form local tissue. And now you might just have a product that would be a material that's very bioactive. So one of the ways we think about and try to learn, how should I design this promoting gel environment, is we think about the native materials in our body, the native extracellular matrix. And so those native extracellular matrix, things like collagen or fibrin, that's our blood clotting, they can contain many different important cues. And that's what is used to recruit when we cut ourselves or cut our skin. The clot that forms has many signals. It's a material that recruits local cells to come in and regenerate and repair the damaged tissue. So when we think about the native extracellular matrix, what we think about is, well, first, many of these soft tissues, like I said, have a high water content and can give some mechanical cues and have a mechanical function. That's what our permissive gels did. But in a simplified sense, some of the things they were missing is the native matrix has adhesion proteins that cells bind to, and they have receptors that will bind to this and give them survival cues. These native matrices also have sites within them that when cells make a certain protease or enzyme, it can degrade this matrix. It can naturally break it down. It doesn't have to hydrolyze, and I engineer it to hydrolyze at a certain rate. The cell decides when I'm going to degrade this. And it also contains systems that can sequester all these different growth factors that can be released on demand. That's really important for healing. So the tricky part, though, with these proteins, though, is that it's very difficult in the lab to isolate these proteins and to reassemble them into gels in a reproducible manner, that looks like and has the functional properties of what they do in the tissue. So we sometimes use peptides as short regions, repeating units in these full proteins. We take just short sequences that are easy to make in the lab and reproducibly that capture a function of this protein. So for example, in these adhesive peptides, we know 3 amino acids, arginine, glycine, and aspartic acid, RGD, is something cells can bind to. And that's the site that they bind, and they'll get a survival cue. This is the sequence that's found in collagen, and cells make a lot of proteases that can cleave this sequence. We also know different sequences that will bind different growth factors, such as transforming growth factor beta, which is bound by this peptide sequence. So what we do now is we take our same favorite PEG molecule, and we just change its end group. And what we do is we change it happens to be a norbornene end group, and we pick this one because this reacts really efficiently with a thiol, and this happens to be an amino acid cysteine. So it's a thiol on cysteine. So now when I go to the lab, I can use an automated peptide synthesizer and make my peptide sequences of interest, and as long as I put a cysteine on it, it can click together and react with my PEG. And so I won't go through, if any of you are interested afterwards, the reaction mechanism by which this occurs, but you mix these two together and they're stable, and it's catalyzed by exposure to light, which I'll come back to why that's important later. So you can click together these hydrogels using these two different components. And the nice thing about that is my PEG part, what's its molecular weight, its number of arms, controls my mesh. That gives me my physical properties in a reproducible way. And the peptide part gives me my biological signals, so I can tune it and put in the signals that are important for my cells. So let me just show you one example where we put those same bone marrow stem cells that in the permissive gels didn't survive and couldn't move. Now put them inside of this type of promoting gel formulation, and first, the adhesive ligand RGD helps them survive long-term in culture. And this degradable sequence here can be cleaved by enzymes that these cells make. So now these cells are sitting inside of this gel, and they are attaching and they're degrading away this matrix locally and allowing them to move. And if you misspell the amino acid sequence so it's no longer degradable, these cells will survive because they have the adhesive ligand, but they can't degrade this very efficiently and move. So it's clearly something where you can tune these different types of sequences to your cell of interest. Now, I put up one more image here, so you can also stain and look at these cells inside of here and compare what they look like compared to their bone marrow niche. But the other reason I put this up is that one of the things that we really need is we have lots of advances in this field of how to track and watch these cells in real time. And we can label cells and know all kinds of things that they're making. But what's hard is we don't know how the gel is being changed. So we need lots of techniques and better techniques to characterize the local changes that are happening to this gel, which is something that I can't see with my optical microscopy. So if you take these gels and you put them inside of a critical size bone defect, so these bone marrow cells can migrate through it. So here in a rat skull, you create a 9mm diameter defect. It's one that can't heal by itself, it's so large. And if you put a hydrogel in place here, what you can't see is that the cells will infiltrate through that. You can see a little bit of mineralized tissue that they're forming here, but it's not very efficient, and it's mostly around the periphery. And that's because even though the cells can migrate into this, they need to get a signal to tell it to regenerate and mineralize and form a bone matrix. So that's where now you can not only design systems that cells can infiltrate, but then you can entrap and present molecules to these cells to give them factors that might be osteoinductive. And there are lots of ways to do this. If your molecule's big and will stay entrapped inside of the gel, then you can just entrap it and it will slowly diffuse out. So that can work pretty well, but if your molecules are smaller that you're trying to deliver, they're gonna come out fast. So then sometimes you have to either tether them or you put them in little microspheres that would slowly degrade over time to release them. And this can be very nice, but then you kind of also worry about, have I destroyed my activity when I tether this in? And another strategy to do, which I'll mention briefly, is you can put in these peptides that will bind these molecules with affinity, and it slows down their release rate and locally sequesters it, much like it's done in the natural extracellular matrix. So that's some of the work that we've done, is we put in different pendant peptides that will bind growth factors with affinity, and depending upon the concentration of the growth factor relative to the strength of the affinity and the number of binders, you can dramatically change and slow down and localize the release of molecules. And so I'll just show you one example now. So if we use this to present or deliver bone morphogenic protein, which is used clinically as an osteoinductive factor to try to fuse vertebrae for people that have problems with their spine. But the problem is with these BMPs, you have to be really careful about localizing that release, because you don't want bone to form elsewhere. And so when we load up that same system and put the BMPs, notice the dramatic differences that you get in bone regeneration. So now you just have a simple material, you're just allowing local cells to infiltrate, and you can promote bone regeneration. But you do have to be careful, because loading and local delivery are really important. So here's the dose that's equivalent to what's delivered clinically in humans, but now for our rat. And this delivery mechanism is pretty efficient, and if I use that high a dose, I get lots of excess bone formation outside of the defect. And if I knock this down by an order of magnitude, I can begin to get something that's much more confined to the defect state and regenerating in a more appropriate manner. But that's something we still need to figure out in this field, is what's the right time scale, the right dose, and the right way to present these molecules to preserve their activity. So the last few minutes, the last 10, 15 minutes, what I'd like to tell you about is sometimes when we're designing these hydrogels and trying to promote tissue regeneration, we need to take a step back. And one of the things that we don't understand are, which signals do I need to incorporate? And sometimes it's more than one signal. And how does a cell process that? And how can I figure this out? And I'd like to give you this in the context of one of our projects that we've been working on, and that's with trying to address the question related to heart valve disease. So in our hearts, we all have 4 valves. They're responsible for directing flow from chamber to chamber. The leaflets in this tissue, it serves as a one-way valve. It's supposed to last our lifetime. But it's a very delicate tissue. It's only 200 to 300 microns in thickness, and it has to constantly remodel and repair. Pressure drops across the aortic valve are tremendous. So many times, different things can happen that this process, where these cells that are supposed to remodel this tissue, something goes wrong. And instead, they form very thick stenotic valves that can't open and close very properly. And if you have a diseased valve that even starts to calcify, the option is a valve replacement. So there are no known ways to reverse this. So we became very interested in, well, why does this happen with these cells? And we wanted to understand, because if we did, maybe we could find a drug that could reverse this. And so one thing, though, what's really tricky about this is when you take these valve cells outside of a valve and try to culture them, what happens is they behave very, very differently in a culture environment as compared to in the tissue. So much different that it becomes really hard to study what might be able to reverse valve disease. So what I'm showing you here is, this is a microarray analysis of all the genes that these cells express. We cluster them together, and here's what happens to the cells that are just freshly taken out of the valve, and here's what happens when you put them on a plate. Now, I'm not going to go into all the genes and what this means, but hopefully by eye you can see there's a tremendous difference in the pattern of this expression just by putting these cells in culture on a tissue culture plate. And in fact, this may or may not surprise you, but thousands of genes go up and thousands of genes go down. But when I show this to my biologist friends, what they find interesting is that they know that transforming growth factor beta is one of the things that's problematic and causes valve disease. That signaling is involved. If you treat these cells with TGF beta, this potent molecule, tens of genes go up, tens of genes go down. And they're different genes, most of them are, than the effect of the environment or the matrix. So the reason I tell you this is that so we became interested. Could we use some of these hydrogel environments as a better culture platform to keep these cells more like they would be in their resident valve-like environment, and use this as a model to study disease and look at reversal of disease? And in fact, these particular genes don't matter, but look at the patterns of these genes. So here's what they are when you just take them out of the valve. Haven't plated them yet. Here's what happens if you never take them out of the valve. And here's what happens if you just put them in one of our soft gel environments. And here's what happens if you just put them on a plastic plate that's normally done for culturing cells. Huge differences. So this isn't perfect, this gel, but we start to recapitulate aspects that are more similar to the cells in the valve. So we got interested in saying, well, what is it about this environment? Is it just a mechanical signal? And perhaps by better recapitulating the valve's mechanical environment, these cells are behaving more like they would in the native valve environment. And so our previous gel systems were ones that these cells would remodel and degrade, and constantly, it allows us to observe cells in a way in which it's more native-like. But we also get interested in dynamic systems that not just the cells dynamically change, but as the experimenter, I can watch cells inside of this gel, and then I would trigger a change around those cells, and then watch how the cells respond. So what if I could soften the environment around the cells and watch how they would respond to see if that might reverse valve disease, which causes stiffening? What if I could soften it somehow and find a drug that would soften it? So that was our question. And we use our same favorite PEG molecule here, but now instead of a hydrolytically degradable group or a peptide degradable group, we put in a chemistry that's light cleavable. So now if I make a gel with this light cleavable group, I can put this gel on my microscope and bring up a laser, and with that light, I can start to break down or cleave the gel and change its properties. And so I'll show you how this looks. You put a gel on a rheometer and you expose it to light while you're simultaneously monitoring the elasticity of the gel. And it starts out stiffer, and then as I expose to light, it cleaves, and it softens and softens and softens. And from this experiment, I can get the kinetics. And it just depends upon the light intensity, the wavelength of the light I use, so how strongly that absorbs. But the neat experiment that one can do is now you can have a dynamic environment where you can culture cells in an environment with a certain stiffness. Exposed to light and in situ soften. So now it'll be half as stiff as it was. And if I shutter the light, it will stay at that new environment, and I can watch how cells respond. And then I can re-expose to light and soften it again to maybe a quarter of its original value. So there are all kinds of unique experiments one could do. And this was the experiment that we wanted to do with the valve cells. We wanted to culture these valve cells on these gels. We had a hypothesis that they were activating to what's called a myofibroblast. That's the cell that's responsible. This activation puts the cell in a repair state. And if it gets stuck in this repair state, it just keeps making tissue and it can't turn off. And a hallmark marker is alpha smooth muscle actin. And that's characteristic of these myofibroblasts. And so here's what happened when you put these cells on tissue culture plasticware. They almost all become these activated myofibroblasts. They're all in the disease state when you're studying them. And they'll make lots of matrix, they'll become highly contractile, and they'll snap together, and they'll even form these calcific nodules that is reminiscent more of what happens in disease progression. So the question that we had was, well, let's use our photolabile gels, and the first thing we wanted to know is, would these cells respond to substrate elasticity? And so first we created a gradient of elasticity across a gel just by passing a mask at a defined rate over the gel surface. And when you do that, it creates a gradient in light exposure and a gradient in the modulus of the material as a function of position. And so these points here, we were measuring the modulus with nano indentation, and this red line is just what we would predict based upon exposure time from our rheometry experiments. Now, if you put the cells on this gradient environment, what we were interested in is there a threshold at which these cells will become activated? And indeed, that's the case. So now blue is the cell nuclei, red is just the cytoskeleton of the cell, and green is that protein alpha smooth muscle actin. So we looked at this in various spots on this gel, and we quantified this. And after 3 days, we found regions where the cells would be highly activated on the stiffer side, almost 80% myofibroblasts, and on the softer side, almost no myofibroblasts. So now we have two different conditions, one where they're in the quiescent state and one where they're in the wound healing activated state. But now here's the interesting question. So we can culture these cells either as myofibroblasts as fibroblasts, but we want to know, can we reverse? Can we turn off the myofibroblasts? And so we did this experiment. We cultured cells in the activating environment, let them become myofibroblasts. Then we took half of our samples and we softened it. And our question was, would they revert? Would they die? Would they apoptose? It wasn't known what the fate of these myofibroblasts were, even in healthy tissues. And so we stained them and looked for the marker of myofibroblasts in the stiff environment. They activate after 3 days, and they stay activated if you leave them in the stiff environment. And then if you take these and soften, what happens is the cells, it's reversible. Softening can cause this reversal and go back to the quiescent fibroblast phenotype, and they behave as if they're always in a soft environment. Again, we can quantify this too and show in our stiff case we get activation and high levels of activation, soft, there are no myofibroblasts, stiff, and we can reverse it by softening. So that's interesting because now we can learn, and we're looking now at the intracellular signaling pathways of what's changing in the cells as it goes in the soft environment, and we're looking at small molecules that can make cells think they're in a soft environment, so they'll go back to a fibroblast with the hopes that this might be able to be delivered and used as a treatment to treat valve disease. So just, where's the future? I mean, these are examples that I've shown you today where we're kind of focusing on and looking at one signal, a mechanical signal, a degradation signal, a protein that's delivered. But the reality is that as we try to tackle more complex problems, we're going to need to be able to deliver many cues in a sequential way. We may not recapitulate everything that the body does when it's healing itself in a proper fashion, but we might be able to figure out what are the key rate-limiting steps and design systems that could intervene in that way. And so the way that we kind of think about this is that thiol-ene polymerization, where we're watching these cells much like the native environment. Well, we think about ways, could I in situ change that environment? And the thiol-ene chemistry is light activated, so that means I can pattern. So I can take cells in this culture environment and introduce new cues and define them in spatially defined manners and introduce them sequentially. I can create gradients using mass or 2-photon or single photon optics. So here's a vision. Culturing cells in an environment that I can watch and observe them, but then simultaneously introduce one signal to certain groups of cells. And then maybe introduce a second signal. So within a given environment, I have cells with no signal, with the red signal, with the green signal, with both signals. And I can watch how those cells respond. And then I can also use my photodegradable chemistry and capture cells. Maybe I'm interested in the cells in this middle region right here. So I saw something interesting. Now I can take a laser light and use almost like capture microdissection and remove those cells. At a later point in time, I can maybe take these cells. And then I can further culture them or analyze them or put them in a new environment. So these are some of the things we think about. We also have a future where it's possible to put cells in an environment and use lasers to erode away channels and create features. And then you can decorate those channels with different chemistries, again using light, to coax cells to grow in certain ways. Maybe you want a neuron to connect up with a muscle, so you'd form a functional synapse. One of the big challenges right now for that field are growing vascular structures, which I think we'll hear about in another keynote talk. So I hope what I've shared with you today is there's lots of advances in material design, and hydrogels are just one example, and they're very versatile systems for delivering molecules to cells, whether in vivo or during culture. they allow us ways to simplify down the extracellular matrix and try to figure out what are some of the key biophysical and biochemical cues that are important. Dynamic environments that allow you to do unique experiments and causes changes are really allowing us to learn a lot about how cells get signals from this environment and how it's processed internally. But there's many challenges, and I think the critical parts are figuring out which cues are important, what's their right context to deliver, and what's the right time scale. And then we always put this in the context of asking ourselves, how simple is complex enough? So I don't need to recapitulate everything, but what are the simple number of cues that introduces enough complexity so I can tackle some of the real challenges? And with that, of course, I'd just like to thank a great group of students, undergraduates, graduate students, visiting scientists, postdocs. Everything that I showed you today, I didn't do a single experiment, so I feel really fortunate to work with a great group of people at the University of Colorado. I include names of many of the key lab members and former members who contributed to aspects of this talk, but I'd also like to recognize some great faculty collaborators, Leslie Lindwan on the heart valve project, Chris Bowman with the peptide functionalized gels, the thiol-ene chemistry, and Jennifer Alicia, we've worked for many years with some of the cartilage regeneration. And with that, I'd be delighted to answer any questions if there are.
And we have time for some questions, I believe, so you can come to the mic.
Hi, Abigail Parks. I've actually worked with Andrea Casco at UCLA. I just got my PhD there. I had a question about, you talked about photopolymerization, but also photodegradation. Can you talk about the advantages and disadvantages of the wavelengths of light you use and how viable that is to using that actually in vivo?
Yeah, that's a great question. So for the photopolymerization, you can form gels in situ with these techniques if there's an open surgery or an orthoscopic technique. It's easy to introduce light under those conditions. The photodegradation, though, is trickier because especially many of the wavelengths of light don't penetrate our body deeply. So one might have to change to something like an infrared sensitive linker, potentially. But I think from my perspective, the value is that there may be niche applications in ophthalmology where you could put something in someone's eye and have a dose release. But I think its true value is it helps you hypothesis test. It allows you to figure out, how did a cell process that signal when I softened the gel or when I cleaved and released a molecule? And then if I can figure that out, then I can design a hydrolytic system or another way to recapitulate that. But in one single system, I can have lots of different delivery profiles, lots of different softening profiles. But I think it helps more with the fundamentals of figuring out matrix signaling and which signals are important.
Another question?
Hi, thank you so much for this talk. My question is, have you all done any work on, and how would you approach if you were trying to, I guess, promote growth of two different cell types or two pretty relatively different expressions of cells, such as when you have bone marrow and your calcified bone in one kind of platform?
Yeah, no, that's a great question. So we using multiple cell types is tricky. So one of the ways that we do think about it is an example that you gave, where you use a single cell type, like a bone marrow stem cell, that can form multiple cells in situ. So then the key and the approach we take is to create a gel environment where in certain regions it promotes osteogenesis, in certain regions it promotes chondrogenesis, so you can recapitulate that interface that exists at the tissue. Others, it becomes more difficult. If you have a tissue that has to be innervated and vascularized, the growth rates and the requirements for the matrices of those tissues and their spatial organization of the cells becomes very important. So I didn't quite give enough of an example, but in that last example that I showed you, what we're thinking about is a system where you might be able to grow myoblasts or muscles in the surrounding gel, but then we use lasers to create patterns to guide neurons to grow in. So we're trying to spatially control a second cell type. And then I guess the third one is I think we're farther along with co-cultures where things like the heart valve work that we did. I showed you some about the valve interstitial cells, which are found in the body of the valve leaflet. But another very important cell type that's important with valve disease are the endothelial cells that line that. And that's a little bit of an easier co-culture model to do because I can make a cell-laden gel with the valve cells, and then I grow an endothelial cell lining around the edge. And it's a thin tissue, so I don't have to worry about a blood supply. It doesn't have a blood supply.
Thank you for a great talk. I'm from Texas Tech University, and my question is related with the leaflet structures. So leaflet structure has two Young's modulus, like a low, the stress, they have like a pretty low slope, is low Young's modulus, and high have like very high, like a steep Young's modulus. And did you thought about like that structure, like it's somehow, biphasic mechanical property, things like that? Can you discuss a little bit more about it, if you have any thought?
Yeah, no, I think that's a good point. And, I think that's an area where there's lots of directions one could take to try to get some of the unique properties, whether it's for a suture or for many tissues have biphasic properties to them. So there's lots of research in creating dual networks. I showed some really simple characterization of mechanics of materials. Some of these can be quite brittle because of network connectivity. And so the way we try to get extensibility is by making more idealized network structures. But then you can imagine making a network inside of a network or trying to introduce some anisotropy. We haven't personally done that, but I know there's lots of work in the field in that direction.
More questions? How much do you feel that your work is measurement limited? And what else would you like to measure? You're measuring lots of things.
Right. No, that's a good point. So how much is our field measurement limited? So this is an area where I think there's lots of opportunity because many of us in our field talk about the importance of culturing cells in 3 dimensions. And obviously, if you want to regrow tissues, you have to grow cells in 3 dimensions. But many of the techniques that we use are disruptive, that I'll have one sample and I will then for each data point, I might have to replicate it 3 times, and I destroy the sample to do my classic biological analysis. So I might measure biochemical content, and I digest away the matrix to do this. Another issue with this is you'd like to be able to in situ measure this and follow things in real time. And I mentioned there's lots of advances right now in probes of cells and what cells are doing in 3 dimensions. But we're really lagging in that what we want to know is what's happening to the gel environment around this. How is the cell changing and remodeling and secreting in this extracellular space? And I think there's lots of advances in maybe microreological techniques and traction force microscopy that allows you to see some of these things in 3 dimension. But I think it could benefit more from non-invasive ways. Can we use different spectroscopic techniques to try to get at this using ultrasound or other imaging techniques? All of these, I think in our field, it's a great opportunity that there's lots of technology that probably could be translated and make quite a big impact.
Any more questions? Let's thank your speaker. Spectacular talk. Thank you.